Once thought lost to the Ice Age, the dire wolf may be prowling the Earth once more. Thanks to Colossal Biosciences, 3 genetically engineered cubs, Romulus, Remus, and Khaleesi, are alive and growing. But are these truly dire wolves reborn, or something far more unsettling? Before we celebrate, there's a darker truth lurking beneath their resurrection. Ben Lamb and George Church made waves when they launched Colossal Biosciences, a startup bent on de-extincting Ice Age beasts. In mid-2024, they announced the birth of 3 dire wolf cubs, Romulus, Remus, and Khaleesi, born in a secure barn under constant video watch. The company says these pups came from gray wolf eggs whose DNA was edited to match fossil bits of the long-gone Canis dirus. Footage shows 5-month-old cubs tumbling over hay bales, chasing one another under skylights, and learning to bark with a sharper howl. Colossal describes this hands-on phase as proof that their edited embryos can develop Ice Age traits in living pups. All gene editing steps, embryo work, and puppy rearing occur under strict biocontainment, in locked labs, climate-controlled barns, and 24/7 video surveillance, to guard against any slip in their de-extinction pipeline. Yet, outside the barn, experts have raised red flags. Many point out that only 14 genes were tweaked in gray wolf DNA, a tiny sliver of the genome. Some call the pups designer wolves, rather than true dire wolves. They argue that appearance alone does not make an animal identical to its Ice Age cousin. Others note that no one has shown fully sequenced dire wolf DNA, only bits from fossils that are too old or too damaged for a full genome map. Wildlife biologists worry these cubs will offer little insight into real dire wolf behavior or ecology. They question whether genetic edits can bring back lost instincts, like group hunting techniques on frozen plains or adaptations to kill giant prey. Ethicists also weigh in. Is it right to craft living showpieces when countless species today need habitat and protection? The dire wolf story began in 1854, when explorers unearthed large jaw fragments and teeth along the Ohio riverbanks. These heavy bones spoke of a mighty canine, far surpassing today's gray wolf in skull size and tooth strength. 4 years later, paleontologist Joseph Leidy gave it the name Canis dirus, literally fearsome dog, placing it in the same genus as modern wolves. Through the late 1800s, paleontologists raced to dig more bones. At sites like the La Brea tar pits in Los Angeles, tens of thousands of dire wolf bones are piled up in sticky asphalt. Wyoming badlands and Texas ranchlands yielded skeletons that filled entire wagons. Researchers carefully cleaned fragments, then pieced together skulls and limb bones to sketch the animal's broad head and robust frame. In 1918, John Campbell Merriam studied dire wolf fossils at La Brea and argued these bones belonged in a new genus. He pointed to unique tooth patterns and limb proportions that set them apart from Canis lupus. His peers, however, stuck with Leidy's classification and shelved Merriam's proposal for decades. By the mid-20th century, reconstructions of the dire wolf appeared in museum halls as giant gray-brown canines with deep muzzles and strong jaws, often posed snarling over a bison carcass. Artists filled gaps in fossils by blending features of modern wolves and big dogs, creating a creature that lived in cold steppes and grasslands across North America. Key dig sites kept expanding our picture. Tar pit bonanzas revealed pack-killing behavior when bones of multiple individuals lay tangled near giant sloth remains. In mid-1900s field journals, amateur diggers noted huge tracks beside fossil beds, tracks too wide and deep for any living wolf. By the turn of the 21st century, researchers had more fossils than ever, but still lacked a clear evolutionary path. Was the dire wolf, a sister lineage, or something wholly unique? As we'll see next, modern genetic work finally traced that ancient family tree and shed new light on Merriam's long-ignored hunch. In 2021, a team published a breakthrough genetic study on dire wolf remains from La Brea. Using advanced sequencing, they reconstructed fragments of DNA dating back up to 50,000 years. To their surprise, the genetic code showed that dire wolves split from the gray wolf and coyote lineage about 5.7 million years ago, long before modern wolves evolved. This evolutionary map places dire wolves as a distinct canid branch, closer to an ancestral dog-wolf group than to any existing species. Fossils and DNA show that during the Pleistocene, dire wolves and gray wolves lived side by side in North America, from Alaska's grasslands to Mexico's river valleys, yet they never interbred. Their genomes contain no sign of hybrid genes. Armed with these findings, scientists revived Merriam's century-old idea of a separate genus. Some now propose naming the dire wolf Enoceandirus to reflect its unique lineage. If accepted, this change would honor the wolf's deep split from the Canis species and cement its place as an Ice Age specialist. For de-extinction efforts, this distinction matters. Recreating a true dire wolf means guiding gene edits toward traits that evolved over millions of years: bite strength, skull shape, and metabolic adaptations for cold climates. Simply editing a dozen genes in a gray wolf may miss countless hidden differences in behavior and physiology. At the same time, the DNA study highlights the value of dire wolf fossils as genetic time capsules. Even fragmented DNA can reveal ancient migrations, population sizes, and extinction timing. Researchers found that dire wolf populations crashed around 13,000 years ago, likely as big prey, like mammoths and giant ground sloths, vanished. This genetic roadmap shows two big challenges for any revival: assembling a full, high-quality genome and understanding how long-lost genes interact in living cells. Without these insights, gene-edited wolves may look the part, but lack the real Ice Age adaptations that defined dire wolves. Next, we'll explore how their bones and fossils paint a living picture of their size, strength, and pack life. Dire wolves were built for power. Their skulls measured up to 40 centimeters long, about 25% larger than gray wolves. Wide cheekbones and deep jaws housed massive teeth capable of cracking large bones. This bone crusher bite let them access marrow, a vital fat source in harsh, cold seasons. Fur color likely ranged from pale cream to light gray, matching frozen plains and snowy ridges. Thick underfur and long guard hairs provided warmth on glacial steppes. Seasonal sheds may have adjusted coat length as climates warmed or cooled over millennia. Bone beds at La Brea and other sites show dire wolves hunted in packs. Side-by-side skeletons lie amid bison and horse remains, suggesting group ambush tactics rather than lone stalking. Print casts near fossil beds reveal paw prints walking 5 abreast, like a wolf team moving in unison. Their homes spanned a vast range: tundra-like ridges in Alaska, grasslands of the Midwest, and brushy river valleys down to central Mexico. They needed open spaces to chase swift prey across frozen ground. Fossil teeth wear patterns point to diets heavy in bison, horse, peccary, and even giant sloth leftovers. Sensory anatomy hints at keen hunting skills. Large nasal cavities suggest a strong sense of smell, crucial for tracking prey over long distances. Inner ear bones imply acute hearing, able to pick up rustling hoofbeats or distant howls in icy air. Social bones, like healed fractures in youth and matching tooth marks on adults, reveal a cooperative pack life. Injured dire wolves survived long enough to heal, likely fed by group members. This care network echoes modern wolf packs, but on a larger scale, tuned to Pleistocene challenges. Together, anatomy and behavior paint dire wolves as apex Ice Age predators. They balanced brute strength with social teamwork, thriving where megafauna ranged. But as large prey dwindled and climates shifted at the end of the last Ice Age, their specialized niche collapsed. Their extinction 13,000 years ago closed a chapter on North America's giant wolf legacy. Today, their fossils and DNA keep that chapter open for science and for the bold dream of bringing them back. Dire wolves roamed North America until about 13,000 years ago, near the close of the last Ice Age. Their final fossils date to this time, marking the end of a lineage that had thrived for hundreds of thousands of years. As glaciers melted and climates warmed, the open steppes and frozen plains they favored gave way to woodlands and brush that did not suit their hunting style. A key driver was the large animal collapse, or megafauna die-off. Dire wolves depended on big prey: bison, horses, camels, and even ground sloths. When many of these giants vanished around 13,000 years ago, a rich food source disappeared almost overnight. Without enough large targets to chase, dire wolves faced hunger and competition. Rapid warming after the last glacial maximum reshaped habitats. Tundra and grasslands shrank as forests expanded. Dire wolves built for wide, cold plains struggled to adapt to denser woods. Meanwhile, smaller, more flexible predators like gray wolves and coyotes moved into mixed forests and open areas, outcompeting dire wolves for scattered food. Genetic bottlenecks also played a part. Dire wolves never interbred with gray wolves or coyotes, so they could not refresh their gene pool. When population numbers fell, harmful mutations piled up and resilience dropped. A limited set of genes meant little room to adapt to fast environmental change or new diseases. Humans arrived in North America around the same time, spreading across the continent in small bands. Some scientists argue that early hunters might have chipped away at dire wolf numbers by chasing shared prey or directly hunting wolves. Yet the scale of human impact remains debated. Most evidence points to climate and prey loss as the main causes. Bringing back an extinct species means one of three main paths: backbreeding, cloning, or genetic engineering. Each has strengths and trade-offs when faced with the ghost of a lost animal like the dire wolf. Backbreeding picks a living relative and breeds individuals that show traits of the extinct species. The Quagga project illustrates this. Zebras with fewer stripes were mated over generations until offspring looked nearly like the vanished quagga. The result resembles the original, but those zebras lack many unknown traits and hidden genes of true quaggas. Backbreeding works best when many descendants still carry key traits in their DNA and when the extinct species had simple features. Cloning and somatic cell nuclear transfer aim to make an exact copy by inserting a nucleus from an extinct cell into an egg of a close relative. The famous Bocardo goat was brought back briefly this way, only to die soon after birth. Cloning demands healthy, intact cells, hard to find in species long gone. Even when cells exist, success rates are low, and animals often suffer health issues or die in early development. Genetic engineering offers precise gene editing using tools like CRISPR-Cas9. Scientists sequence fragments of fossil DNA, compare them to a living kin for dire wolves, gray wolves, and identify key gene differences: size, skull shape, coat color, metabolism. Then they edit those genes into living cells, grow embryos, and implant them in surrogate mothers. This method promises traits closer to the original species but hinges on having a fairly complete genome map. Fossil DNA is often damaged and incomplete, leaving gaps in knowledge. All methods face surrogacy challenges. Marsupials, like the Tasmanian tiger, need pouch technologies. Birds, like the dodo, require egg manipulation, and large mammals demand safe pregnancy durations. For dire wolves, using dog surrogates simplified some steps, but raised welfare questions about mismatched womb environments. Colossal Biosciences laid out a clear protocol to resurrect the dire wolf, blending fossil DNA and gene editing. Here's how they say it unfolded. DNA sourcing. Researchers secured two prime fossils, a 13,000-year-old tooth from Ohio's Sheridan Cave and a 70,000-year-old skull from Idaho's American Falls Reservoir. Both yielded fragments of ancient DNA, enough to identify gene sequences linked to size, skull shape, and coat traits. Genome assembly. Lab teams stitched these fragments into a partial dire wolf reference genome. Though far from complete, this map held 500 times more data than prior studies. Scientists compared it to the gray wolf genome, finding 99.5% similarity, but pinpointing 14 key differences tied to physical features. Target gene selection. From the assembled data, they chose 14 loci, spots on the genome influencing body size, head width, fur color, and hair length. These genes, once edited, should express traits matching Ice Age packs rather than modern forest wolves. CRISPR-Cas9 editing. Using the genetic scissors tool, millions of gray wolf cells had their DNA cut and replaced at the 14 target points. Lab cultures grew thousands of edited cells, each a tiny test of how changes might manifest in living tissue. Embryo creation. Edited cell nuclei were inserted into dog egg cells, stripped of their original nuclei. The eggs developed into embryos in vitro. Out of 45 attempts, only 2 embryos matured far enough to implant. Surrogate implantation. 2 embryos found homes in domestic dog surrogates. Experts opted for a C-section to reduce risks. Dire wolf pups could grow larger than dog litters. On October 1st, 2024, Romulus and Remus arrived safely. Twin development. Though genetically identical, the pups showed distinct personalities. One boldly explored new toys, the other paused and assessed before joining in. Care teams tracked growth, behavior, and health under constant watch. Third pup. A later embryo, following the same process, produced Khaleesi on January 30th, 2025. She added a female presence to the small pack in a high-security barn. Romulus, Remus, and Khaleesi live under constant monitoring in a secure barn. Though their DNA echoes Ice Age hunters, their early life follows a human script more than a wild pack's. Surrogate rearing. From birth, dog mothers provided initial care, but staff soon added bottle feedings, balancing mother's milk with formula tailored to dire wolf metabolism. Growth charts track weight, height, and skull dimensions, comparing them closely to fossil predictions. Early behavior. Despite shared DNA, the twins diverged. One pup raced after toys, the other hung back, sniffing corners before joining play. These differences underline how the environment shapes personality. Even clones can develop unique traits based on subtle cues from caregivers, lighting, and handling routines. Health monitoring. Veterinary teams perform daily checks: temperature, heart rate, joint flexibility, and tooth growth. Special attention goes to hip development and spine alignment, since large-headed pups risk joint strain. Fur texture and coloration also get notes, matching expectations for pale, dense coats meant for cold climates. Social structure. The trio bonds with each other and with handlers, but lacks older dire wolf mentors. Without wild pack dynamics, leaders, hunters, pups, researchers wonder if true social instincts will emerge. To simulate pack life, staff introduce puzzle feeders and scent trails, encouraging teamwork and finding hidden snacks. Breeding limits. Colossal plans only 3 to 5 more litters before halting reproduction. Security fences and drones prevent escapes but also isolate the pups from natural stimuli. Their world remains a blend of hay bales, heated dens, and rotation through fenced yards. Quality of life questions. Can a barn-raised animal fulfill its Ice Age destiny? Critics argue that these cubs become living museum pieces, lacking true freedom and purpose. Supporters say they'll gain knowledge for the future and more natural reserves. Either way, these wolves will never roam the tundra or hunt bison. Ethical balance. As pups grow, questions loom. Will physical traits alone define them, or will they need rigorous training and space to test Ice Age instincts? In controlled care, staff strive to mimic seasonal changes, cooler nights, raw meat hunts, to spark wild behaviors. Yet ultimate choice rests with humans. These cubs stand at the edge of extinction revival, caught between a barn and their ancient heritage. Can these new, dire wolves ever fill the roles their Ice Age ancestors held? Today, parks and preserves rely on large hunters to balance deer, elk, and bison. Dire wolves hunted giant herbivores long gone. If released now, they might compete fiercely with gray wolves for the same prey. More top predators could leave too few animals for all of them. Space is another issue. North America's wildlands have shrunk. Many reserves barely support existing wolf packs. Adding dire wolves risks upsetting fragile food chains. Could they chase off foxes and coyotes? Might they carry diseases or stray onto farms? We simply don't know how a revived dire wolf would fit into today's wildlife. On the moral side, is it right to spend millions on creating a show crew of extinct animals when living species teeter on the edge? Northern white rhinos, pangolins, and island birds need habitat and anti-poaching efforts now. Their survival depends on real-world conservation, not gene labs. Should our best science protect what remains instead of chasing the lost? Yet some experts argue that bringing back dire wolves could restore lost ecosystem services. Big hunters shape plant life by culling weak prey, clearing brush, and helping seeds spread. If gray wolves alone no longer fill that role, a blend of gray and dire traits might revive long-gone rhythms in grasslands and forests. And this may just be step one. Colossal Biosciences is already planning woolly mammoths, Tasmanian tigers, and dodo birds as their next de-extinction targets. Each project will test our genetic tools and raise fresh questions. Where would these giants live? What duties do we owe them and the ecosystems they once shaped? As labs push to resurrect more species, we face big choices. Do we use bold gene science to rewrite extinction's final chapter? Or do we focus on saving the species still with us, guarding them against real threats today? The coming years will show whether de-extinction stays a lab wonder or becomes a force for rebuilding wild balance. From ancient bone beds to cutting-edge gene labs, the dire wolf's return forces us to rethink extinction. Bringing back a top predator sparks hope, wonder, and tough questions about our priorities. Will these engineered wolves roam wild lands again or stay as curiosities behind high fences?